Hi everyone, and welcome back to Genealogy and Decoding Your Y-DNA, a series about how Y-DNA testing can help us explore our paternal lines. My name is Jeremy Lehman, and I am a genealogist who loves weaving DNA into family history research. In this video, we'll take a look into how ancient DNA is transforming our understanding of human history. Let's first discuss what ancient DNA is. Ancient DNA of humans refers to the genetic material recovered from the remains of humans that lived in the past, anywhere from hundreds to tens of thousands of years ago. The defining characteristic of ancient DNA is that it is fragmented and chemically damaged, which makes it difficult to retrieve and sequence. It doesn't necessarily mean that the DNA dates back to ancient times. The ability to retrieve ancient DNA from human specimens sounds like science fiction, but there have been remarkable advances in extraction and sequencing technology over the past 15 years. So much so that today we now have ancient DNA samples from over 10,000 humans, sequenced from all populated continents, and as far back as 50,000 years ago. This has been no small feat. The work involved in retrieving ancient DNA is orders of magnitude more difficult than the samples that we living humans send to the lab with our saliva or cheek swabs. Scientists have to search for small fragments of DNA preserved in areas such as the petrous bone behind the back of the ear. And they have to contend with significant contamination from outside sources, mostly from microorganisms, but also from modern humans like ourselves. There are still more hurdles to overcome, especially when it comes to retrieving ancient DNA from warm and wet environments. But the advances made so far have ushered in a revolution in the study of ancient DNA. There has been a flood of ancient DNA papers just in the past few years. While ancient DNA research largely focuses on autosomal DNA, there usually is also information about the male specimen's Y-DNA haplogroups. And this is exciting for Y-DNA researchers. These Y-DNA haplogroups can help date particular haplogroups to specific times and places in the past. And in turn, this information is enhancing our understanding of human history. Let's discuss two examples of how important ancient DNA research is incorporating Y-DNA in their analyses. Perhaps the seminal scientific article that ignited the ancient DNA frenzy was a paper back in 2015 that came from David Reich's lab at Harvard. In that paper, the study looked at the ancient DNA of 69 Europeans, dating from 8,000 years ago to 3,000 years ago. And what they discovered was astonishing. They showed that the Yamnaya, a group of pastoralists from the Pontic steppe in Ukraine and Russia, were involved in a mass migration that significantly contributed to the ancestry of Europeans in the cultures that followed. And this has continued on to the present day, where Europeans, especially those from Northern Europe, continue to have a significant amount of Yamnaya ancestry in their DNA. Y-DNA was a central part of the paper's analysis, They studied 7 male ancient DNA samples from the Yamnaya and showed that they all belong to the RM269 Y-DNA haplogroup with an R1b. RM269 was practically non-existent in European samples before the time of the Yamnaya. But in the centuries that followed, it would become the dominant lineage in Europe, and remain so today. The Y-DNA evidence showed that the spread of RM269 and its downstream subclads in Europe only began after 3000 BCE and was mediated by the migration of men from the steppe. Now, fast forward 10 years to today. While we now have a refined understanding of steppe paternal lineages, there are still lingering questions. The Yamnaya males have been shown to largely be part of the RZ2103 subclad of RM269. But later cultures in Europe, such as the corded ware and bell beaker cultures, showed greater diversity of Y-DNA steppe lineages. These included the RL51 subclad, which is a separate R1b lineage within RM269, as well as the RM417 subclad that is within R1a. We still aren't sure of these latter lineages' precise origins, but those Y-DNA haplogroups became the dominant steppe lineages in modern Europeans today. Hopefully, future research will shed more light about how they emerged and spread through the European population. Another example of the transformative nature of ancient DNA relates to the history of the Ashkenazi Jewish population in Europe. Back in 2022, a paper was published about a medieval Jewish community in Erfurt, Germany. Scientists investigated 38 skeletal remains from a Jewish cemetery in Erfurt dating to the 1300s and 1400s, and they found something very interesting. The ancient DNA revealed 2 distinct subgroups of Jews in the community. One that appeared closer to Middle Eastern populations, which they termed Erfurt ME, and another that was closer to European populations, which they termed Erfurt EU. Modern Ashkenazi Jews with Eastern European ancestry appear to be a mixture of these 2 populations, whereas those with Western European ancestry are largely from the Middle Eastern-like population. Their research suggested that the Ashkenazi population in Erfurt descended from 2 distinct populations. One, Erfurt ME, that may have come from the early Rhineland Ashkenazi communities that were to the west of Erfurt. And a second, Erfurt EU, that came from areas further east of Erfurt, in the Czech lands of Bohemia, Moravia, and Silesia. This latter Erfurt EU group appears to have had admixture with the local European population to a much more significant degree than what was observed for the Rhineland Ashkenazi community further west. Interestingly, the evidence of these 2 origins of Ashkenazi communities theorized in the Erfurt paper is also supported by linguistic, cultural, and onomastic studies that have shown differences between Ashkenazi Jews from Western Europe and those from Central and Eastern Europe. The paper also presented 10 Y-DNA lineages of the male samples in Erfurt that were studied. Most of the 10 lineages are present today in the modern-day Ashkenazi population, which shows the continuity of these lines through the centuries, and attests to their existence in Europe going back at least 600 to 700 years. And in fact, 3 of these lineages exist in my own ancestry, which was exciting for me to discover. This means that these 3 Erfurt men who died in the 1300s and 1400s were paternal line relatives with members of my family. Perhaps even more intriguing, though, was the male sample's assignment into the 2 Erfurt subgroups. Haplogroups that were associated with Erfurt ME men appear to be associated with lineages that have deep roots in the Rhineland. A good example of this is the man from the JL556 lineage, a prominent Ashkenazi lineage today that has the highest percentage of modern-day testers with paternal line roots in Western Europe. And the haplogroups associated with Erfurt EU men appear to be associated with lineages with lower representation in Western Europe. A good example of this is the man from EY6940 lineage, which is one of the largest Ashkenazi branches in the modern-day population, but of which very few individuals have paternal line roots in Western Europe. Thus, the Y-DNA evidence from the male samples from Erfurt hints at additional support for the theory of 2 distinct Erfurt subgroups with different histories of migration. This is an exciting development and hopefully can be followed up on in subsequent ancient DNA studies. Hopefully, these 2 examples illustrate how exciting ancient DNA research can be. So how can you learn more about these ancient DNA samples and their associated Y-DNA lineages? The easiest way to learn more about the Y-DNA lines from ancient DNA is through Family Tree DNA's Discover page. The Ancient Connections tab will provide you with a list of ancient DNA samples that are most closely connected with a particular haplogroup. Ancient Connections will also provide a date for when the sample shares a most recent common ancestor with a haplogroup, and will provide a citation for the study in which the sample was analyzed. The best part about Ancient Connections is that it is freely available for any Y-DNA haplogroup. Just type in a haplogroup of interest and you'll see their closest ancient DNA connections. If you want to dive in deeper, I would recommend consulting with the Allen Ancient DNA Resource, better known as the AADR. The AADR is an initiative from David Reich's lab at Harvard University, meant to serve as a central resource for the more than 10,000 published ancient DNA samples. The AADR incorporates samples from hundreds of ancient DNA papers and helps curate the data in an analyzable format. It is periodically updated and it is free for anyone to download the data. Researchers from the University of Richmond have built a tool incorporating the AADR dataset called the AADR Visualizer. It provides a geographic visualization of all the ancient DNA samples in the AADR database and allows you to easily filter through the data and export it into a spreadsheet. For example, with the AADR Visualizer, you can filter for specific Y-DNA haplogroups, or you can filter for a particular study, so you can learn more about the ancient DNA samples found in a specific time and location. It's a great resource worth checking out. The landscape of ancient DNA continues to evolve quickly, putting it on the cutting edge of scientific discovery. Our understanding of human history has been transformed over the past decade of research, and is poised for further discoveries in the coming years. Y-DNA lineages play an important role in these studies and in turn help us better understand our own paternal lineages in the modern day. Have you come across any interesting ancient DNA studies? And who are your closest ancient DNA, Y-DNA connections? I'd love to hear about them down in the comments. And if you're enjoying this series, be sure to like and subscribe. Let's keep decoding your Y-DNA together.